function session. I'm Callum MacGregor from Newcastle University. I'm going to be chairing. Just a quick couple of things before we launch into the first talk. Is our fifth speaker, LVFSD, in here yet? No, okay, hopefully they'll turn up. For all the speakers, it's the same as every other session. 13 minutes, I'll wave at you when 5 minutes left, and I'll wave at you again when you've got 1 minute left. There's a clock there if you're not sure how well you're doing. And if you're having your talk recorded, if you haven't opted out of having your talk recorded, please try and stay somewhere near the mic so that it gets picked up. So without any further ado, should we go on to our first talk, which is Sarah Faye Harpenslager, I'm doing my best, who's talking about it's getting hot in here, the effects of increasing temperatures on key processes of the carbon capture cycle. Sarah.
Thank you, Callum. You did quite well. It's Harpenslager, which is nice Dutch tea, which nobody else can pronounce anyway. I am a postdoc working at Queen Mary University on the Ring of Fire project. I'm not sure if some of you may have seen other talks by colleagues from this project. We're looking at the effects of warming on a whole ecosystem, like including food webs, microbial communities, and in my case, nutrient cycling. So the Ring of Fire project is called Ring of Fire because we visit sites around the Arctic Circle in Alaska, Kamchatka, Svalbard, Iceland, and Greenland. And these systems consist of stream networks that are located in geothermal areas. So all of these streams are basically similar in physical and chemical properties, but because of the geothermal activity, some of them are warmer than others, and you get a range of about 2 to 40 degrees of temperature. And we use this as a kind of natural laboratory system to study the effects of warming. And we use that in addition to some more artificial heating experiments in mesocosm settings. So this is an extreme example of part of one of these stream systems. I don't think I have to tell you which one of these two streams is a warm one, but apart from the obvious differences in primary production, these systems, because of temperature, are also expected to differ in processes like respiration and in processes involved in the carbon and nitrogen cycle. So both of these cycles, basically they start when nitrogen or CO2 is fixed into biomass, and both of these cycles have end products that are greenhouse gases. So N2O, methane, and CO2 are all products coming from these processes. And with warming, we can here see that primary production increases, and we can expect that most processes will increase depending on the rate of temperature. So all of these processes would then be enhanced, but they won't all be, they're not all dependent on temperature in the same way. Some processes will be enhanced more than others. Some of this work has already been done in warming experiments. For example, this is a Dorset pond experiment, which is a collection of mesocosms, half of which have been warmed about 4 degrees above ambient for the last 10 years. And from these systems, it has been found that because of warming, these red bars here are the warmed ones, and the black bars are the ambient ones. You can see that the ratio between ecosystem respiration and gross primary production changes. So you get a higher ecosystem respiration relative to your primary production, which means that the net carbon sequestration rate has dropped by about 13%. And that's not all. It's also that the proportion of this primary production that is emitted as methane increases. So you get both of these processes as a result of increased temperature because of the temperature dependence of these processes. However, most of this has been done now in these mesocosms, but to see whether this actually happens in the field and to link it up with the nitrogen cycle, we had research goals to look at how these key processes from these two cycles respond to warming in a field setting, looking at these geothermal systems. And we can use this to predict how major biogeochemical cycles will be affected by climate change. Furthermore, because both of these cycles produce greenhouse gases, we can see how warming affects the emission of these gases as kind of a feedback of global warming. And furthermore, we want to see if these temperature dependence of the different processes differ within the cycling of carbon and nitrogen. So for that, we went to the field last summer, and we will go again next summer. And we measured a range of different processes using different approaches. For example, we had oxygen loggers in all streams that gave us Diel curves of oxygen throughout the 24 hours. And we can use this to estimate global gross primary production and ecosystem respiration rates. Then in most of the streams we had closed chambers measuring carbon fluxes, and we had benthic chambers measuring the oxygen consumption of the benthic community, where we expected this slope to be steeper when streams were warmer than when streams were a colder temperature. So here is we see in the stream itself we have probes that we use to measure denitrification in the field itself. So we have probes in the sediment that we inject labeled 15 nitrate into, and then measure the production of 30, like 15 N2 over time. Where in the background, Jill Welter is going to talk later, is measuring N2 fixation, which in these very limited systems is a very important process. And then finally, we used incubations. So we got thousands of these little vials that we fill with sediment and water from the streams. And we add one of these stable isotope substrates. And then we see over time how these substrates are turned over by means of denitrification, nitrification, anamox, methane oxidation, and methanogenesis. And we incubate them in the stream itself so that they're at the temperature that they're actually originating from. So this summer we visited 3 sites. We visited Iceland, Alaska, and Greenland, which apart from being very pretty, they're also really limited in nutrient concentrations. They have very low availability of nitrogen, phosphorus, and carbon in the surface water. And this may help explain some of the other results that I'm going to show you in a minute. So we had oxygen loggers in all streams, giving us Diel oxygen curves. And although I haven't had time to do the estimation yet for primary production ecosystem respiration, since we only got back last September, we can see that the amplitude of these oxygen concentrations during the day differ between a cold stream and a warm stream, which will also later be reflected in the difference in primary production ecosystem respiration. But we will use these processes as a kind of a normalization for the other processes that we measure in these streams. And there is a recent paper by Benoit Demar who shows these processes across a whole range of temperature, and we hope that these actually kind of match up to those. So some data that I do have analyzed by now is the benthic respiration rates. We can see that there is not very strong, but there is a relationship with temperature from all 3 sites that we visited this summer. I'm expecting that especially the organic matter content of these sediments will have a bigger explanatory value, power to it. But at least we can see that there is an effect of temperature here with the respiration. And the same can be said for methane production in these streams. There is a slight increase with temperature. This is that hot stream that I showed in the beginning with lots of primary production. But all other rates are generally, if you compare them to other systems, and I come from peatland systems, so they're very, very low. And the oxidation rates show kind of similar pattern to the methane production. And these rates have actually been obtained by getting higher concentrations, up to 100 times, 50 to 100 times higher concentrations to background methane concentrations here. So these rates will in the field really in situ be limited by substrate. But potentially, because of these rates are quite high, you can see that they would consume almost all the methane that will be produced in these streams. And finally, I've got some nitrogen removal rates from the system. We can see that there is a bit more activity in Alaska, which can be explained by these systems were a bit richer in organic matter and nitrogen because they were covered by, there was more vegetation around these, and there was probably more organic matter flowing into these streams. But again, you see a nice relationship with temperature. Still, if we compare those rates to the Dorset pond rates that I showed earlier, they're like 20 times lower. So you can see that they're really, really substrate limited in these Arctic streams. So some first thoughts based on these results here is that they're extremely low nutrient systems, and all nutrients are probably held within the biomass, not really freely available in the system. We do get some temperature dependence rates, but they are strongly limited by substrate. And we hope to see this same relationship with the functional gene abundance that Kate Randall from Essex University is measuring in the same streams. And we hope to link this to that. So that gives me something to do for the rest of the year. I want to calculate temperature dependence for all the processes that we've measured in the field and finish the analysis. Also, I want to see if we can estimate metabolic processes from the Diel curves and compare the rates that we found to the gene abundance that University of Essex has found in the same streams. Hopefully, I'll be able to manage all of that before we go on the next field trip to Svalbard and Kamchatka in summer next year. And with that, I wanted to thank everybody from the Ring of Fire project and you all for listening. Thank you.
Thank you, Sarah. Exactly. Very nicely to time. Does anyone have any questions for Sarah?
You showed a lot of what I think were mean values. How much variance was there around this?
Differed for the most streams. There was some variance, but there was different per process. I mean, especially some of the hot streams, and it would also depend on where in the stream you took your samples. I had replicates along the stream, and in some points you get very fine sediment that shows, for instance, higher denitrification rates, and if you get the bit gravelly bit on the other side. So there was some variation, but I didn't put them in yet, no. That's right.
Anyone else with a question? Yes.
Are you measuring only complete denitrification, the whole process, or other minerals?
For now, I'm measuring the whole denitrification. I inject them with 50 nitrate, and I look at the production of N2. But we are going to look at also, like, see if we can establish what DNRA and Animox are adding to the process, if that's what you mean.
Brilliant, thank you very much, Sarah.
Thank
I think
you.
we'll move on to the next speaker. The next talk early, in case anyone's coming from other sessions. So just chat amongst yourselves for a couple of minutes, and we'll get our next speaker ready. You've still got a minute and a half. Oh, we've got another question. So we'll move straight on with the next speaker, who you will have read is Sophie Flack from Edinburgh, and she's going to be telling us about carbon economics of LAI drive photosynthesis.
Yeah, unfortunately I don't have any nice fieldwork pictures because I'm a modeller, so I don't go outside. So I'm Sophie Flack, I'm from the University of Edinburgh Global Change Ecology Lab, and this research was conducted in partnership with the Global Ecosystems Monitoring Network. And today I'm going to be talking about how and why leaf area index drives photosynthesis across Amazon precipitation gradients. So tropical rainforests cover 5% of the Earth's land surface, but account for 1/3 of total terrestrial photosynthesis. This makes them an integral part of the global carbon cycle, and understanding drivers of carbon dynamics in these systems is increasingly important. So we know that precipitation is a really important driver of carbon dynamics in these systems, and this graph, adapted from Marley et al. 2015, shows maximum climatological water deficit against gross primary productivity, which is carbon entering the system through photosynthesis. Each of these individual data points is a permanent sample plot across the GEM network, and they are biometrically derived estimates of GPP. And what we can clearly see is that as precipitation regime becomes increasingly dry, gross primary productivity declines. And this is a pretty bog standard, everyone kind of knows that photosynthesis declines with precipitation. So we know that there's this empirical relationship, but what I'm really interested in is how forest response to precipitation influences these carbon dynamics. So to fill in this knowledge gap, I pose the following research questions. First, I want to know what is the principal driver of spatial variation in GPP across this precipitation gradient. So essentially, what a forest doing that affects this interaction. And secondly, I want to understand, well, why do we actually observe this spatial variation? What's driving the driver? So to start off with, answering the first question, I started to consider the potential limitations to gross primary production. And I'll just take you through some of the key biotic pathways and then explain how I arrived at my hypothesis. So the first one, which people tend to always think of with water limitation, is stomatal conductance. But actually, we expected that stomatal conductance would be more of a short than long-term response to precipitation. And actually, if these forests are living in these climates, then they should adapt over time. The next one is nutrient availability, influencing the photosynthetic dark reactions. So potentially, there could be some impact of water availability on nitrogen or phosphorus cycling. But actually, the differences here are more east to west Amazon rather than the equatorial forest down to the dry south. Next is the photosynthetic capacity of leaves. But actually, studies have shown that this has the opposite trend and increases with dryness. So that wouldn't really explain what we're seeing. And so lastly, we arrive at leaf area index. So this is leaf area in metre squared per ground metre squared. And it defines the surface area for potential photosynthesis. Across the Amazon, it increases with precipitation, which, given the evidence of the other drivers, we hypothesised that this was the principal driver of spatial variation. So next, we want to understand why. So it costs carbon to grow leaves, and it costs carbon to maintain them through respiration. So to invest in leaves, it has to be worthwhile, and the increases in photosynthesis need to pay off those costs. So we hypothesised that the drier the site, the less the GPP potential is lower, so this limits the increase that you can get from increasing your leaf area index, and that's what's driving the trends that we see. So, as I said, I'm an ecosystem modeller, so I applied an ecosystem model to 8 permanent sample plots across the Amazon. These are gem network plots. So you can see we've got some in the wetter regions and then some getting drier. They span a mean annual precipitation range of about 1,000 millimetres a year. The soils differ from east to west. There are 2 plots at each of these 4 locations, but the soil and species composition differences avoid any pseudo-replication. So I took data from these sites. I used meteorological data, soil properties, vegetation properties, leaf area index, and leaf traits, and input them to my model. I then validated the model against carbon flux data collected at these sites through the GEM network. So I used the soil plant atmosphere model, SPAR, and I'll just give you a brief overview of the general structure of the model. So we estimate GPP through a series of process-based equations based on the Farquhar model. GPP is then allocated, the carbon assimilated through GPP is then allocated to autotrophic respiration, and this is also calculated independently. The remaining carbon is then allocated between the plant components, and then we have litterfall and so on. So the inputs to this model are time series data of meteorology and leaf area index, together with fixed values of vegetation properties, which includes leaf traits, and also soil properties. The outputs are then carbon stocks and fluxes, as well as water and energy fluxes. So we initially calibrated the model at site conditions. Then to answer our first question, looking at what is the principal driver of variation in GPP, we took our sites that were calibrated, and then we changed the leaf area index. So we essentially changed the model input and then look at the effect on GPP afterwards. We do this at each site with each leaf area index and then repeat the whole thing for vegetation, meteorology and soil. At this stage, we don't allow feedbacks on carbon pools because essentially we don't want the dynamics to get in the way. We want to calculate the direct effects. We then apportion the variance to the individual drivers. To then answer the question of why we observe this spatial variation, we allow the feedbacks, we allow the model to stabilise, and then we can really look at the impacts on carbon dynamics. So, answering our first question, it was leaf area index, you were right. And that accounted for 49% of the variation we see in gross primary productivity across this precipitation gradient. Meteorology was, and vegetation actually surprisingly only accounted for about 5%. A point to point out here as well is that year is included. This just accounts for the fact that we ran the model from 2009 to 2010. So there is some interannual variability with the leaf area index and the meteorology. So next, we want to really look at what we're observing in the field and compare that to what happens across model parameter space. So what we have at the bottom is the forest conditions. So here we have the forests, which include the soil properties, vegetation properties, and meteorological data. We characterise this here by the mean annual precipitation, but it's just a note to point out that this data does include leaf traits, soil, etc. Then on the y-axis, we have mean annual leaf area index. The contours are GPP and measured in grams of carbon per metre squared per year. And the individual data points are the forest conditions under the observed leaf area index time series that was taken from the field measurements. And the error bars are the sample error of leaf area index. So what we can see in general is that as mean annual precipitation increases, so too does GPP, except we have this kind of anomalous little loop here, and this is driven by the fact that these are western sites, and this is eastern Amazon, and essentially the leaves in this area have got higher photosynthetic capacitance, so that's just drawing a little anomaly there. But overall, as mean annual precipitation increases, the minimum LAI required to achieve the maximum potential GPP increases. And if we look at the observed data, so 6 out of the 8 plots fall within the maximum potential GPP range. So we can kind of conclude that the GPP is driven through optimality. However, we do have one site which doesn't fit this trend. What we do notice about this site is that at this location, the GPP is least sensitive to leaf area index. Further investigation showed that these plots were actually in the highest optimal carbon use efficiency. So it's perhaps some kind of risk strategy that we're observing. I just skipped through that one. So why are these findings important? Well, some people have looked at the carbon economics of leaf area index before, but they tend to have looked on a satellite large scale, and they haven't taken into account these really detailed plant traits that we've been able to use in this study. And I think it's been really interesting to, instead of always using a model to predict, rather using it to diagnose what's actually happening in this ecosystem. Really interestingly, so our aim was to look at the spatial variation, but actually a lot of parallels can be drawn with temporal variation in rainfall. So a number of through-fall exclusion experiments have recorded a drop in leaf area index after the onset of the exclusion. And Nepsad et al. 2002 reported that this drop was actually due to a decline in leaf production rather than an increase in leaf turnover. So it wasn't due to increased environmental stress affecting the leaves, but rather a lower production rate suggesting a plant-level response to this change in rainfall. So I think that we're really at the stage now as well where we can start thinking about actually inputting these mechanisms and these theories into our models, so we can really start to look at what could be the potential implication for changes in precipitation across the Amazon, which is changing and is a huge threat to carbon dynamics in this system. And a kind of sad thought as well is that I found it really interesting that it was leaf area index specifically that came out. It's a relatively fast pathway to respond to change. And I think that maybe places it in a higher viewpoint in terms of we need to think about it more, looking at resilience of these forests in the future. So just to conclude, leaf area index is the principal driver of GPP across this Amazonian precipitation gradient. This was driven by the balance between the carbon cost of leaf, construction and maintenance versus photosynthetic potential. And I feel that this research really improves our understanding of the interactions between precipitation, forest feedbacks and carbon dynamics. Thank you very much, and just a special thanks to my supervisors and also everyone that went outside and collected data so that I could use it. Thanks.
Brilliant, thanks very much, Sophie. Any questions for... There we go.
We had several presentations today and yesterday that clearly showed that the tropical forest is primarily nitrogen limited. So what makes you believe that it is carbon limited? This is the underlying assumption of your...
I think that it depends what kind of spatial scale that you're looking at in terms of if it's all nitrogen limited, then, well, yeah, but if you have one area that's nutrient limited, let's say, but then other variables are still going to affect carbon dynamics across that system. So if it's all nutrient limited, then, and actually comparing the east and west Amazon, there wasn't as clear a split as across the precipitation gradient.
I think the logic is just the other way around. If it's more humid, nutrients are more available and you have more frost, and then you have more proteins.
So we would expect that differences in nutrients would be reflected in the leaf traits, but emerging from the model, that wasn't evident.
Any further talk amongst yourselves for 1 minute? Steady influx of people rushing from other sessions, so I think maybe we should just move straight on. Our next speaker is Ruth Mitchell from JHI in Scotland, who's going to be talking about decline in atmospheric sulphur deposition and changes in climate.
Well, good afternoon. Thank you very much for staying on for the last few talks. And today I want to talk to you about some results from a long-term survey we've been doing, looking at a resurvey of grasslands in Scotland and trying to disentangle what those drivers might be of changes in our Scottish grasslands. So just to give you a bit of background, in the 1940s there were two botanists in Scotland called Eric Burse and Jim Robinson, and they had the aim of surveying the Scottish grasslands, or Scottish vegetation. So they had a great job. They spent sort of 30 years wandering around Scotland, recording the vegetation and trying to classify the vegetation and record typical community compositions in Scotland. So they ended up with almost 7,000 records of vegetation in Scotland and the composition of those communities. And this was actually published as the Plant Communities of Scotland in 1980. So at the James Hutton Institute, we've been very fortunate in having these records, and this enables us to go back to them and to look at long-term change. So I'm particularly going to look at the grasslands, and my colleague Andrea after me will be talking about the springs. So the original survey for the grasslands, the plots were recorded between 1958 and 1987. So that gives us an average date of 1973. So I'll refer to these plots as 1973. We then went back and resurveyed these plots between 2012 and 2014. So that neatly gives us an average of 40 years later. And in total, we surveyed 561 plots. So these 561 plots covered a range of different grassland types. And we classified them into 6 grassland types: acid, calcareous, lowland, mesotrophic, nardus and wet grasslands. And this was based on the vegetation composition in the original survey. And we wanted to be able to use this resurvey to see how our grassland composition had changed over this 40-year period. We wanted to identify what the drivers were that were associated with these changes, and then see if there were differences between the different grassland types. So a few results before we go on to look at the drivers, looking at the changes in the species composition. The next few graphs have all got the same style. So the white bars are the results from the 1973 survey, and the black bars are the ones from the more recent resurvey. And when we looked at species richness, we've actually got an increase in species richness in our acid, calcareous and our nardus grasslands. If we look at diversity, using the Shannon Diversity Index, diversity declines in the majority of our grassland types. So the acid, the lowland, the mesotrophic and the wet grasslands saw a decline in diversity. The other index that we looked at was this dominance index, the Berg-Parker Dominance Index. And for those of you that aren't familiar with it, this index looks at how dominant species are changing. So our dominant species getting more dominant. And in all of our different grassland communities, dominant species were shown to become more dominant. And this might initially come across as a bit of a contradiction. We've got species richness increasing, yet at the same time we've got diversity declining. And that's explained by this increase in dominance. So some species are still just about present and other species are coming in, but the diversity overall is declining because we've got this increase in dominance. And this indicates that an extinction debt might be occurring. We haven't actually lost those species yet, but they're declining in abundance as a few species are becoming more dominant. Looking at some of the changes in the different species groups, forb cover generally decreased in the majority of in all of our different grassland types, whereas grasses showed different responses. In the lowland grasslands, which were our most intensively managed grasslands, we had an increase in the abundance of grass cover, whereas in the nardus grasslands, we actually saw a decline. We then wanted to go on and relate this to environmental changes that had occurred over the 40 years. So I thought just briefly, for any people that weren't familiar with the changes in that period in the UK, I'd just run through the broad changes that we've seen over that 40-year period. So looking at the road tap report, NOx emissions have declined quite significantly in that 40-year period, and that's resulted in a 25% or 24% decline in wet deposition. If we look at NHY emissions, they peaked in the 1980s, so just at the end of their first survey, and since then there's been a decrease, but that hasn't actually resulted in a decrease in the deposition. The big one that's changed over the course between that resurvey is SOx, is sulphur. This peaked in the 1970s, so at the start of that survey, the first survey, there were quite high levels of sulphur deposition, but that wet deposition has decreased by about 70% across the whole of the UK by 2010. There's also been changes in climate, so going to Pacific Scottish level data, we've seen increases in mean winter, spring, summer and winter rainfall, as well as an increase in temperature. In terms of grazing, we actually missed the peak livestock numbers. So livestock numbers continued to increase after the original survey. They reached a peak in the 1990s and then decreased after the 1990s when the headage payment was decoupled as part of the agri-environment schemes. So those are the sort of broad changes we've seen, and we wanted to see if we could relate that to our plots and the vegetation. So the drivers we actually put into our model and our analysis, we used the 10-year cumulative data in the pollution variables prior to each of the 2 survey dates. And when we got the data for our plots, or for the locations, and divided it up by the different habitat types, again, you've got the same patterns of SOX decreasing, NHY increasing, and NOx declining. For the climate data, we used the 10-year mean prior to the survey, and we've seen this increase in rain and minimum and maximum monthly temperature. And then the livestock units showed a mixed effect. So livestock units are a combined effect of cattle and sheep, and they increased on the lowland grasslands but decreased on the Nadas grasslands. And the reason we used either the 10-year cumulative or the mean was to try and average out fluctuations in the years preceding the actual survey date, and to try and capture the drivers that were impacting on our survey. So looking at some analysis, we then split those into the 5 different groups that you can see there. We used generalised linear models to look at changes in cover against the changes in drivers. And we used mixed models to look at the effects of species richness. And we tested the effect of each of these drivers once we'd taken into account the effects of our other drivers. So I'm now going to show you the results from this, and these all follow a very similar pattern. I've got my 5 species groups down the side here, my 6 different grassland types across the top, and for each grassland, we split it into whether species richness changes or whether the cover changes. And if there's an increase, then the relevant box is coloured yellow, and if there's a decrease, then the relevant box is coloured blue. So when we're looking at the impact of a decrease in sulphur, you can see that bryophytes actually increased in cover and species richness in 2 of the grasslands, and the Cyperaceae, the sedges and the forbs also increased in the calcareous grasslands in terms of species richness. But there's actually a decline in cover for the grasses in terms of sulphur. So perhaps we're beginning to see here the first indications of an impact of that decline in sulphur emissions. If we look at rainfall, this had a positive impact. When there were significant impacts, everything increased in either cover or species richness, other than for the grasses. So there was no impact in terms of the grasses in any of the different communities. In terms of maximum temperature, an increase in maximum temperature, this had a mixed impact depending on the grassland type and the species group you were looking at. If we look at the results for an increase in minimum temperature, this generally had a negative impact. So generally, species richness or cover, if there was an impact, it resulted in a decline, but the results were more positive for calcareous grasslands. So the sedges and the forbs, that their species richness increased with increasing minimum temperature. So in summary, we've identified this possible extinction debt for our Scottish grasslands, and I guess the policy question here is, can we manage for it? We haven't actually lost those species yet, so what can we do to actually maintain them before that extinction debt kicks in? We're actually perhaps starting to see the first impacts of that decline in sulphur. So that's taken a long time before we've actually seen that impact. Sulphur deposition started to decline in the 1970s, and it's taken us 40 years before we've seen the impact of that policy change. And I think that's a real key message to get across to policymakers, that sometimes we need to wait a long time before we see these impacts. Carly Stevens was talking yesterday about nitrogen impacts and trying to find data to support the fact that these policy changes are having an impact. We've also seen changes in terms of climate, and climate, particularly these increases in temperature and rainfall, do seem to be driving a lot of these changes in our Scottish grasslands. We did also get some results in terms of nitrogen deposition and grazing impacts, but these weren't as consistent. I didn't present them today, but if you want to know more details, do contact me. So finally, it just remains for me to say thank you very much to all the surveyors that did the field work for us, for Jackie who helped with the statistical analysis, and the landowners who gave their permission to access the land. I also need to thank the Scottish Government who funded this work, and you for all listening. So thank you very much.
Thanks very much, Ruth. Does anyone have any questions for Ruth? Lots of questions. Good.
Too much time.
Up the back.
I think it's hard to disentangle exactly which one of those might be causing the extinction debt, because you've got all three of those different things happening in terms of changes in species richness and changes in diversity and dominance, and it could be a mixture between them all. I think the easy one to manage might be the change in grazing, because we have seen quite a decline in grazing across a lot of our grasslands in Scotland recently, and almost sort of abandonment as the grazing's retreated. And certainly looking at the grazing results, it was the more intensively managed grasslands where grazing was causing a decline, and actually a decline in grazing was having an impact on some of the more less intensively managed grasslands. So in terms of which one's easier to manage, to stop that extinction debt occurring, it would be the grazing. But whether that's what's actually causing it is probably a different matter.
Mike.
You averaged your climate data over 10 years.
Yeah.
I just wonder though if that's hiding any effects of extreme years or like a run of cold winters or something like that.
It could be. It's a question of how many different variables you can fit into your model. Yeah.
You've got spatially explicit data, and presumably some of the changes over time were of different magnitude. So can you put multiple variables into a regression to tease out which of these drivers are more important?
Yes, we could do. Yes. Like to tease it out, which all the drivers were in the model.
Simultaneously?
Yes. So all the drivers were in the model, but we took account. The model only tested if any particular driver was significant, having taken account of the effects of all the other drivers. So yeah.
Thanks very much, Ruth. I think we should move on, but we're going to stay with James Hutton Institute and stay with Scotland. Andrea Britton is going to tell us about upland spring communities.
Okay, so I'm going to tell you a bit something about upland springs. This is another piece of work from the Burson-Robertson archive resurvey, so it's very similar to the work that Ruth was just talking about. Okay, so our uplands are changing. We have multiple drivers of change. Over the last 40 years, we've had, as Ruth was describing, we've had a peak and decline in sulfur deposition. We've had nitrogen deposition, which has increased and is now relatively stable. We've also had a peak and decline in grazing impacts. And also now the climate is changing and we're seeing changes in temperature and rainfall. So a whole mixture of different impacts which can be affecting the condition of our upland vegetation. In this particular presentation, I'm going to focus on upland springs. So why are we interested in this particular habitat? Upland springs represent biodiversity hotspots in the upland landscape. They have a very diverse and specialised biota, both flora and fauna. And because the soils remain unfrozen throughout the year, they're very often important feeding areas for upland birds. And these communities as well are also thought to be sensitive to the temperature and quality of the irrigating groundwater. So, looking at these habitats, how might they have responded to the decades of pollutant deposition, grazing impacts, and the recent climate change that we've been seeing? So, the work I'm going to talk about today, it's, as Ruth was describing with the grassland data, it's a long-term resurvey study. It's based on vegetation data which was collected in the 1970s by Eric Burse and Jim Robertson as part of the effort to describe the plant communities of Scotland. We have this huge database, and from that we've selected, in this case, 48 plots which represent a fairly closely related group of spring communities, oligotrophic springs dominated by Philanotis fontana. So in 2008, we relocated 48 plots and resurveyed them. And now we're going to look at how those plots have changed over the last sort of approximately 30 years. So our headline results, overall, we found an increase in species richness between the two surveys, so similar to the grasslands. And in particular, this increase in species richness was driven by an increase in the richness of bryophytes and also graminoids. When we look at the cover of the different groups, we can see increase in graminoid cover, which is a signal which is quite common to a lot of vegetation survey work, which is large-scale vegetation surveys that have been carried out in the UK. We also see declines in the bryophyte and forb cover. So thinking about which species are actually changing the most, these increasing graminoids tend to be common and widespread species that are ubiquitous across a wide range of upland habitats. So we have things such as Nardus stricta, Anthoxantha moderatum and Carex nigra, which have all increased. And when we look at the declining species, a lot of them are the things that are very typical to these particular communities. So we have the bryophytes such as Dichodontium palustri and Philanotis fontana, which are both very typical spring species, and also a range of the forbs, which are very characteristic, things like Pacaltha palustris, Chrysoplenium oppositifolium, Montia fontana, and Saxifraga stellaris. We also looked at changes in the Ellenberg scores to give us some idea of how the changes in species group abundance that we were seeing might relate through to changes in the environment. So we looked at the mean cover-weighted Ellenberg scores for light, for moisture, nitrogen, and reaction. And of those, we can see we have significant change in moisture, nitrogen, and reaction. So the moisture and reaction ones are indicative of the plant community moving more towards drier and more acid conditions. And very interesting, we see this decrease in the nitrogen scores, which suggests a decrease in nitrogen availability, or perhaps overall fertility, which is a better description of the actual Ellenberg indicator score. So it's interesting to consider why that might be, given that nitrogen deposition has increased. Why would we see a decrease in the nitrogen scores in these spring habitats? One possible theory we have here is that that might be an interaction with acidification, and that acidification may have caused a reduction in the availability of some of the nutrients in these spring communities, so things like phosphorus, which can become less available with acidification. So now to go on to more directly analyse these data with the changes in the actual drivers to try and pick out some of the relationships. So similar to how Ruth did with the grassland data, here I'm just going to describe the changes in the drivers for the 10-year periods prior to each survey. So for this set of plots that we looked at, the changes we have, cumulative pollution, so the cumulative sulfur deposition has declined by about 64%, so that's been a really big change. We've also seen a decline in nitrous oxide deposition, but a small increase, significant increase in reduced nitrogen deposition. On the grazing front, we have quite a large significant increase in deer density, but when we look at the sheep, so the domestic stock, we don't see any overall change, but that actually masks quite a lot of variability between the plots in different locations. So we have some that have seen a big increase in grazing, some have seen a big decline, but overall there hasn't been a change in sheep density across all of the plots. And then when we look at the climate, we've had a 15% increase in the amount of growing degree days. We have an small increase in summer rainfall and a much larger increase in winter rainfall. So we would anticipate that any of these things could be impacting on the spring communities. So to relate these changes in drivers to the changes in vegetation change, the approach we took was to use best subsets regression to look at the relationships between the vegetation change metrics and potential drivers. For the richness change, we used GLM analyses. And when we were looking at the pollution variables in this particular habitat, we decided we were interested both in the change in the actual deposition rate, which has generally been a decline for, let's say, sulfur dioxide, but we were also interested in the effect of the ongoing accumulation of pollutants between the two surveys, because you might expect different responses to the two. So in our analyses, we included both the 10-year mean prior to the survey period and also the accumulated deposition since 1850. So I'm going to show you a few tables here which are quite similar. So what we have on these is along the top, these are our driver change metrics here. So we start with pollutions. This is the change in pollution prior to the survey date. This is the cumulative pollution between the survey dates. Here we have the grazing, change in grazing, and the change in climate variables. And then the metrics down the side here. So this is change again. So we're looking at change versus change. And in all these cases, positive relationships are shown by the red squares and negative relationships by the blue squares. So hopefully that'll be fairly easy enough to follow. Okay, so first of all, looking at the Ellenberg scores, we see that the declining sulfur deposition is related, we have a negative relationship here with the reaction score. So that's showing that declining sulfur deposition is associated with reduced acidity. So that's perhaps some indication of recovery from acidification impacts. When we look at the cumulative NOx deposition, you can see that seems to be associated with increased nitrogen availability and also increased acidity. So that's perhaps, again, suggesting an ongoing impact of NOx deposition, increasing the nitrogen availability and also more acidification still ongoing. We didn't see many signs of significant relationships between climate and changes in moisture status, which we thought we might have done. We thought that, changes in winter rainfall might have made the community moisture scores increase, but we haven't seen any evidence of that. Okay, looking at changes in species group cover. So in this one, we see evidence for declining sulfur deposition. I'm going to get the right one. Yeah, so declining sulfur deposition associated with increased forb and bryophyte cover. So again, that's perhaps some more evidence that we might be seeing recovery from sulfur deposition impacts. And also we see cumulative sulfur deposition associated with a reduction in bryophyte cover. So that perhaps is evidence of an ongoing impact of sulfur deposition, even though we've had such a large reduction, there seems to be some ongoing impact. And then a result here which is in common with a lot of other studies from a range of other habitats, we see that cumulative reduced nitrogen deposition is associated with increased graminoid cover. So again, an ongoing impact of nitrogen deposition. And an interesting positive effect of sheep grazing is that we saw that sheep grazing here, so reduction in sheep grazing, I guess right now, continuing sheep grazing is associated with reduced graminoid cover and increased bryophyte cover. So it perhaps suggests that some level of grazing is beneficial to actually maintain these communities. Okay, and finally, a response to increasing growing degree days, we see an increase in graminoid cover and a reduction in forb cover. So finally, coming on to look at species richness. Here we see, again, a result which has been brought out by a number of other studies. So increasing reduced nitrogen deposition is associated with a decline in species richness, especially in this case in graminoids and forbs had a particularly large impact. Interestingly, we saw a small positive effect of cumulative reduced nitrogen deposition. This was much smaller than the impact of the actual change in reduced nitrogen deposition between the two surveys, and we're not quite sure why that might be, but it does show up again in the same groups, particularly for the graminoids and forbs. And again, we see a positive effect of grazing on species richness, in that sheep grazing is associated with higher richness across a whole range of groups. And in terms of climate change impacts, we see that summer rainfall is associated with reduced forb richness. Okay. So just to summarise the sort of key points that emerge from the resurvey, we're seeing significant change in these spring habitats over a period of 3 decades. We're seeing increased richness, which is associated with an increase in ubiquitous species and a decline in specialist species, although very few species have actually gone extinct. So again, similarly to the grasslands, it raises this issue of perhaps an extinction debt beginning to occur. We have complex driver-vegetation change relationships, so all of the drivers that we looked at were associated with vegetation change in some way. So it just emphasises the complexity of the situation. In terms of impacts, we're seeing continuing impacts of acidification, although sulphur deposition has declined a lot, and we did see reduced impact where sulphur deposition had declined the most, but there are still continuing effects of acidification. And these are contributed to both by sulphur, but also by NOx, which contributes to both acidification and eutrophication. And in terms of the impacts of reduced nitrogen, we're mainly seeing these in terms of reduced richness and increases in graminoid cover, which, as I said, was a result which has been seen across a wide range of studies in the UK. And we are beginning to see deleterious impacts of warming, so increases in graminoids, reduction in forbs, which are both changes which reduce the sort of special character of these particular habitats. But interestingly, we do have this beneficial impact of grazing in that it seems to be able to maintain the open nature of some of the spring communities and perhaps prevent the ingress of grass species. Okay, so it just remains to me to thank Scottish Government for funding and also DEFRA and our statistician and landowners for helping with the project, and also to thank Eric Burse and Jim Robertson for having done the original data collection and having the foresight not to throw anything away. And thank you all very much for listening.
Thank you very much, Andrew. We've got time for one very quick question.
The missing on the unmarked indicators is that they are interpolate within the species. So you shouldn't do 6 separate independent tests, but maybe some permutation test, which accounts for this, which help in disentangle which driver is behind.
Yeah, I mean, we could do that. At the moment, we've tested them independently. So we've looked at each particular metric. But yes, there are a lot of interactions between them.
So the reaction and the nutrient indicators are really strongly correlated.
Yeah, yeah.
Okay, thank you very much, Andrea. We'll move on to our last speaker in this session, who is Benthic, who's going to be telling us about relationships between geomorphic disturbance dynamics and vegetation change in the Alps.
Hopefully. Okay. So, I am an Italian PhD student and I will present you this part of my project. As you all know, we are seeing impact of climate change on these environmental factors, so air temperature, precipitation, greenhouse gases, etc. And they're having in turn impacts on terrestrial ecosystems. But this could mean both having impacts on geomorphic processes and in turn on biota system, and vice versa, having impacts on biota systems and in turn giving impacts on geomorphic processes. So why it's important to study these biogeomorphic processes? Well, there is a gap of knowledge in these ecological and geomorphic processes. And then there are few studies, especially on periglacial systems. And yes, it's important to figure out the climate change, especially in the vegetation communities, and other kind of feedbacks on climate change, like CO2 concentration or redistribution of carbon. Where to study these biogeomorphic processes? Well, it's important in areas where climate is changing rapidly, like alpine ecosystems. And then in environment where the conditions are varying rapidly, like exposure or soil moisture, so the Alps, and where we found unexpected impacts on vegetation driven by geomorphic dynamics. For example, Stelvio Park, where I work. How to study them? There are several ways. First of all, the fieldwork method. Then you can collect a lot of proxies in different fields, like the speed of movements of landforms, the soil type, the vegetation cover. Then you can put everything into a GIS and evaluate the differencing between these factors. Then you can pass to a statistics model. But it's also important a new field, the photogrammetry, that is the structure from motion subfield of photogrammetry. So my aims is to assess and quantify the biogeomorphic processes at different temporal and spatial scale in Stelvio Park, combine diverse methods to obtain larger and reliable data set, and also associate the surface disturbance with their geomorphic genetic processes like creep, hydrological processes, gravitation processes, and salifluction. Here is my fieldwork site in Central Alps near Bormio. And this slide is very important because, first of all, we started by producing two vegetation maps, one from 1953 and the other one from 2003. And we translated the vegetation communities into three ecological types: the pioneers communities, the climax communities, and the bare ground. And by a simple operation in GIS environment, we got these colored areas. And for example, if you see the light green one is we have a change from bare ground communities to pioneers communities. And so thanks to that, I was able to select the points, the more interesting area in my fieldwork site. Which kind of landform we studied? First of all, periglacial landforms typical on those areas, like salifluction lobes, and terracets, ploughing blocks, and ice needles. They are typical small landforms. Then we studied also cryotic landforms, typical of permafrost environment, so rock glaciers and block streams, and also gravitation or hydrological landforms like debris flow and ski slopes. So, for example, for salifluction lobes, terracets, and all the small landforms, we used to study the morphometry of the landforms. Then we used the diastimeter quadrat to get a height profile of the landform. We put vegetational releves. We did a 3D model using the photogrammetry technique and also soil excavation. For bigger landforms, the main method was to paint transversal line along these landforms and to check the vegetation lying on this painted line at each centimeter. So we got an information about the vegetation, and also by surveying the old lines, we were able to understand the intensity of movements of the rocks falling from this line. this is not very interesting, it's just an overall characteristics of the landforms. So the first results are, for example, for Rock Glacier 1, we have the faster movements in the front lines, so the front of the rock glacier, and practically no movements in the upper part. But this is a long-term monitoring, so we resurveyed the old lines. And this is the vegetation lying on this rock glacier. You can see that we have an high occurrence of Ranunculus glacialis and Leucanthemopsis alpina and Po alpina. And they are divided into colors. Green is pioneer species, blue is snowbed species, and yellow is grassland and shrubland species. For Rock Glacier 2, we have a different situation, so we did not record movements for the middle lines like this one and this one, but only on lines laying on the front, so on the two scarps of this rock glacier. And again, we can find a high occurrence of Ranunculus glacialis, Leucanthemopsis alpina, and Po alpina. This is a stone-banked lobe next to the Rock Glacier 2, and it has a similar movement to the previous Rock Glacier, so with a high velocity at the front here and slow velocity on the upper part. And the vegetation is again pretty much similar with Leucanthemopsis, Po alpina, and Ranunculus glacialis. Then we passed to the scree slopes on the other side of the mountain, and here we faced a totally different movement. So in this case, the upper lines are moving faster than the lower lines, as you can see. And we have also a high occurrence of other species like Gnaphalium supinum, Sedum alpestre, Festuca alleri. So there is a different vegetation. On the scree slope 2, in another site, it's a long-term monitoring. We registered the same movement, but with a lot of intensity for the uppermost line. This is the vegetation of scree slope 2, pretty much similar to the previous one. And we also studied debris flow, and we divided it into the scree and the proper debris flow. And you can see only two species are tolerating the movement of this debris flow, so Sedum alpestre and Festuca alleri. So one main important thing is that the overall vegetation I studied is linked to these community types, but only the first three ones are the ones who tolerate the movements of stones. And if we have a look into these three communities, we can see that there is also a high species turnover during time from these three different periods. In fact, you can see for Oxyretum digine, there is a lot of turnover percentage, and even more for Androsacetum alpine during the years. And there is a strange trend for Luzuletum spadice, so first a decrease and then an increase of other species. And what we would like to do is to have this graph, so have a look at this column. This is the total presence of each single species in relation to the movement. So these are the records. For example, if we take in consideration Leontodon helveticus, okay, there are few records, so we will improve the number of records, but every time I surveyed it, it's in correlation with a movement of stones. And in contrast, if we consider Saxifraga bryoides, I register its presence only in 14.3% of movements. So basically, it stays where the movements are not occurring. And in conclusion, so we have different dynamics for each landform type, but we have also different landforms with similar movements. And so maybe there is a background process producing this kind of landform. For example, the gravitation and not only the creep of permafrost. And then for the long-term monitoring, we registered a high species turnover and an increase of species richness, but with low frequencies. So there might be geomorphic disturbance preventing the upward migration of lowland species. Then, of course, pioneer species are tolerating landform dynamics more than other species. And lastly, among tolerating species, we have different strategies. So we have consolidator species of fine material, like Poalpina and Leucanthemopsis alpina, and associated species to them living in sub-niches, so Veronica alpina and Saxifraga gabrioides, and coarse material tolerating species like Ranunculus glacialis. And that's it. Thank you for listening.
Brilliant, thank you, Stefano. Are there any questions for Stefano? I have a question. I hope it's not a silly one. You talked a bit about climate change and how that might interact with geomorphic processes. Do you think that's likely to have any sort of negative effects on any particular groups of species in terms of their tolerance to movement?
Yes, basically we are experiencing a movement of lowland species towards the more elevation sites, but with climate change, so we are expecting a totally shift of lowland species, but this movement is prevented by these kind of dynamics processes. So I think it's positive to have these processes, but we have to study more to be sure on which species are tolerating these movements and which are not.
Brilliant. Thank you, Stefano. Thank you again to all of our speakers in this session for keeping to time and for keeping us entertained throughout the graveyard slot of the last session of the conference.